Good morning, and I'd like to welcome everyone to our BioDigital event, Getting Prepared for BioDigital, and our BC Showcase event. My name is Ryan Butta. I'm the Director of Partnerships and Events with Life Sciences BC. Thrilled to welcome everybody from around the world to our event today. You're in for a real treat and lots of great presentations. Just some housekeeping before we get started is that a reminder about the chat function. Please put any of your thoughts, comments, great feedback in there. Also, if you look at the top right, you'll see 4 little arrows. When you click on that, that allows you to actually go into full screen mode. In the Q&A section, you can put in any questions and you can upvote to highlight a popular question. Please note there's not going to be any live Q&A, but all of the speakers will be able to go into the Q&A section and answer your questions after their presentations. After all of our presentations, we're going to be having a networking session, and there's going to have 2 8-minute sessions. So you can move to the front of the room, double click on an empty chair to move from table to table, and then introduce yourself for 1 minute before moving on to another table. But we would ask that you please wait for the 8 minutes before you move to another table, just to avoid any disruption. And when we go into the networking, please make sure you turn your camera and microphone on so that other attendees can see you. And you can also upload your picture and social media links to make it easier to network. So without further ado, I'd like to kick off the program by introducing Wendy Hurlburt, who's our President and CEO. Over to you, Wendy.
Hello, everyone. As Ryan said, my name is Wendy Hurlburt, and I'm very excited to be here this year at our 2nd annual BC Showcase at BioDigital. As we begin, I'd like to acknowledge that I'm joining this event from within the ancestral, traditional, and unceded territory of the Coast Salish peoples, including the territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. I'd also like to acknowledge that others are joining us from traditional and unceded Indigenous territories from across this beautiful province and country. So welcome to today's webinar, and thank you for joining us. We're very excited to be here, and we really appreciate you taking the time to join us to learn about BC innovation. Over the past year, our community has embraced virtual events, and we've been thrilled to be able to tour the world virtually, essentially, and meet with many companies and investors and potential partners for our BC-based companies. This event is going to showcase amazing innovation taking place in BC, and you'll hear from 9 companies who are going to be attending the conference. As well, we have a booth in the global marketplace that we hope you'll visit us throughout the conference. A little bit about Life Sciences BC. We're a not-for-profit organization, and our members consist of academia, research institutions, centers of excellence, funding agencies, emerging small, medium, and growing digital health, medtech, biotech, and many global companies operating in that space. We also have many partners that are members who specifically dedicate their services to the life sciences sector. For example, lawyers, regulatory consultants, privacy consultants, compliance, accountants. So opportunities in BC and British Columbia, please check out our website where you'll learn about who those potential partners can be. Next slide, please. Our event is made possible by our event sponsors, Ferris and Silicon Valley Bank, and our event supporter, the British Columbia government. Next slide, please. So I thought I would just take a moment, since I know that more than half of you that are in attendance are from outside of BC, and also more than half of you are not members of Life Sciences BC. So I thought I would take a moment to just give you a little bit of an overview on this sector before we actually move into the company presentations. So British Columbia is home to some of Canada's largest life sciences companies. Stem Cell Technologies is the largest biotech employer in Canada, and we have 5 companies greater than $1 billion in market cap based in BC: AbCellera, Aurinia, more that are coming up to that milestone. I know of at least 5 or 6 that are getting close to that. The life sciences sector raises about $5.4 billion in revenue. This is based off of 2018 data, and clearly we've seen a lot of growth over the last year, so probably even higher. 1.6 billion in GDP, 500 million in exports. We're the fastest growing life sciences sector in Canada and one of the fastest growing sectors within British Columbia. There are 2,000 registered life sciences companies in British Columbia, and that's growing at a rate of about 5%. We have a highly skilled workforce, approximately 20,000 employees, and our emerging workforce of 22,000 students studying in the life sciences related fields. That's growing at about 15% and probably one of the fastest growing fields of students' academics. As well, we raised $2 billion of capital and brought that into the province in 2019-20. Next slide, please. So what is BC known for? Well, we've got some very strong world-class clusters that BC is known for, and I'm just going to mention a few of them. Antibodies, regenerative and cellular medicine, nanomedicine, lipid nanoparticles. We often say we're good at anything small, or we make anything small big. Oncology, genomics-based precision medicine and gene therapies. We've got a very strong medtech sector with medical device design, prosthesis, UV and infrared equipment, a number of companies operating in the 3D printing space. Digital health. One of the things that sets BC apart is that we're very strong at creating solutions for human health, which combine physics, biology, chemistry as an engineered solution. So the scientific disciplines to create solutions is one of our great strengths. We are a point of connection between human health, agriculture, food science, and clean tech, and we increasingly see companies emerging that overlap in those subsectors. And we have a very, very strong talent pool with a very knowledge-driven culture and sector. Next slide, please. So I thought I'd touch it wouldn't be 2021 if I didn't say something about COVID. So when necessity struck, we really saw the BC life sciences ecosystem come together and shine globally. We've been recognized for our world-class research. We showed we can act fast. All of our innovation was grounded, of course, in science-grounded innovation, but I think we saw that people generally saw the importance of this science-grounded innovation, which really helped bolster the biotech sector generally. We showed that we can pivot. We demonstrated the value of global collaboration and partnerships, something that Canada is very strong with. And we showed that all stakeholders working together, research, industry, government, regulators, collaborators, we saw what we can accomplish. Next slide, please. So we like to believe that BC innovation really punched above our weight. And many of you probably are aware of this, from antibody discovery to mRNA vaccines, ventilators, digital health. BC companies really led the way here. Our local Abcelera partnered with Lilly on developing the therapeutic. The Pfizer vaccine is, we like to refer to it as powered by ACUTE technology, with that being the delivery mechanism. Precision nanosystems has been very active in this space with vaccines and their products being used globally. Starfish Medical pivoted very quickly into the ventilator space, as did Triumph, and Thrive Health became the basic mainstay digital health tool. So lots of innovation coming out of BC, and as you can see, it had a lot to do with collaboration, global partnerships, acting fast, and building off of our world-class research. So we hope you'll come visit us at the BC booth in the Canadian Pavilion. Listed here are all the BC-based companies that are in attendance at BIO, so please look them up. And you're going to hear from a number of them over the next little while. Turn it over. I'd like to introduce James Hatton from Ferris to say a few words, who has been supporting our presence at this conference. Over to you, James. James, you're on mute.
Somebody had to go first. Thanks very much, Wendy. And I'm going to steal a line you were using today. We make anything small big. I'm going to say at Ferris, we help make anything small big. On behalf of Ferris, my partner Hector Mackay-Dunn, who's with us here today, and myself, I'd like to welcome everyone again to the BC Showcase for Biodigital. We at Ferris are longtime supporters of Life Sciences BC and the life sciences in BC. Hector and I both cut our teeth in life sciences, and it's been our chosen practice area our entire careers. Hector got me involved in my very first life sciences file with my first client, which was QLT. And at the time in BC, QLT was the only life sciences company. The little known fact is that Hector was acting for them at the time because he'd acted against QLT when they went public. And they said to themselves, next time we're doing something like this, let's have that guy on our side instead of against us. My job here today is to say a few words and then stand back. I kind of wish I was speaking at the end, because I was looking at this fantastic lineup of speakers, and today we're going to be hearing from some old friends and some new ones, and some people whose story I'm hearing for the first time. But I always come away from these Life Sciences BC events flushed with enthusiasm. It's the story of the people and their quests. It's so motivating, it's so inspiring. You just can't help but be proud and excited. There's just no better industry to be a part of. And we're all doing our best to be doing well by doing good. At Ferris, we've been with you, many of you, all along the way, and we in your journey, and we celebrate your victories, we share your defeats. I think of us as your Sherpas, urging you to greater heights. And I like to think we play a small part in your successes. We thank you for your confidence in us and for giving us that opportunity. And now it's time for me to stand back and hear the stories we all came here for. Thank you.
Thank you very much, James. And I would say Ferris has been a huge supporter of this sector and has always been a supporter of our BC delegation to Bio. So it's great to have you as part of this one as well. Okay, we're going to start to move into the presentations, and our first speaker is Anne Woods. She's the Managing Director of the Life Science and Healthcare Practice for Silicon Valley Bank in Canada. She's based in Toronto. She's a huge, passionate supporter of life sciences. And we asked her to join us today so she can give a little bit of an overview of setting the stage of what's happening with investments and capital attraction in British Columbia and Canada in our sector. So over to you, Anne. Thanks.
Thanks, Wendy. You may be asking yourselves, why is a born and raised Torontonian who sits on the board of Life Sciences Ontario and works for an American company presenting on BC? But I think that's really a testament to what Wendy said earlier is, BC is drawing a lot of attention, not just within Canada, but from outside of Canada, and really is punching above its weight. And one of the things that Silicon Valley Bank is known for is our life science sector reports. And, consistently over the last few years, when we're looking at sort of some of the, great success stories, whether it's from Series A investments or to exits, we consistently see BC companies popping up there. So as a Torontonian, I guess I can say I much prefer losing to Vancouver than to Montreal. And of course, that's a bit of a sore spot for us. So I'm not going to dive too much into the global sort of details. I think I'm probably preaching to the choir in this crowd if I talk about the record amounts of capital raised globally, or the amount invested in companies, globally and in Canada. You can see all that information in our reports, and I'll post a link in the chat to them. But what I thought I would do is just really give a bit of an overview about what we're seeing in BC and why we're so excited about it. So Wendy mentioned the $2 billion in capital that's raised, and that amount has been sort of going up year over year for at least the last 4 years. It is the fastest growing sector in BC when we look at the number of employees and company creation, growing faster than both Ontario and Quebec. And the life sciences sector in BC attracts an outsized amount of venture capital dollars. So when you look at the total amount of venture dollars invested in BC, 40% of that goes into life science companies versus only 15% in the rest of Canada. So lots of great numbers that are really sort of highlighting the current state. But then you start diving into the companies. And so there's some great companies that are going to be presenting today. And then you've got companies that Wendy mentioned earlier. So there's Aurinia and CHINOOK and Xenon and Zymeworks and ESSA. And if nothing else, think about just the number of lives that have been saved in the last 15 months due to technology that came out of BC companies. So of course, ACUTE and AbCellera developing the therapeutic, but then not to mention the sort of, work that stem cell technologies and precision nanosystems have also done kind of, behind the scenes, supporting all the research that's happening around the world. And it's not just biotech. So we've got Canary Medical, so a medical device company that was named one of Fierce MedTech's 15 companies to watch over 2021. A real game-changing technology for patients with implantable medical devices. And then, of course, Aspect Biosystems, which humbly calls itself a 3D printing company, but in reality is combining microfluidics with 3D bioprinting to create living human tissue. So a real sort of amazing technology. And so, why has BC created this sort of, amazing depth and breadth of technology? So I think UBC is a big reason for that. If you ever get a chance to see a presentation by the ACUTE team, you can see the global impact of the nanomedicines research group at UBC that goes all the way back to the late 70s. I also think that the proximity to the US West Coast and access to both capital and talent that exists on that coast has really resulted in an ability to tap into that talent. So when we couldn't find it here at home, we could just look a short distance away to Seattle to get some of that executive talent to help grow our companies. And I think a really important piece is the entire community is really all about collaboration versus competition. And so you can attend any number of events and you'll see executives and team members from all companies working together to grow the ecosystem. And I think that's a real unique feature in the life sciences sector, but you can really see it living out in BC. So why are we at Silicon Valley Bank really bullish on BC? We've got 2 employees there now, and we're continuing to expand in the West. And really, the momentum is amazing. So on the heels of some huge 2020 IPOs, we just saw last week an announcement from Precision Nanosystems that they've been acquired by Danaher. And what is amazing about all of this is these companies are not getting acquired or raising huge amounts of capital and then packing up and leaving BC. They're doubling down and investing in building manufacturing and hiring hundreds and hundreds of employees. And so it's really a good sign that this momentum is going to continue, and that all of these companies that are being developed are going to continue to spin out talent to start new companies. And it's just going to keep building, and you'll get this real snowball effect. We're seeing, of course, big interest from both the federal and provincial governments. And so whether it's around policy or financing, the sector, of course, is attracting a lot of attention. There's even talent, if I think of Notch Therapeutics that's in Toronto, they didn't look to Boston for executive talent. They looked to BC. And so I think that's a big step forward too, that the broader country is really recognizing BC. And I think finally, the most important piece is mindset. So there's been a real mindset change and an attitude shift that entrepreneurs and investors are willing to take that risk now. So we've got so many success stories that new entrepreneurs have that vision of what it can be like for them, and I think that's creating a lot more risk takers and a really great environment. And I think all the gears are really working together for future success in BC. And so with that, let's get to the presentation so you can really hear about the science and listen to some of the fantastic entrepreneurs. Back to you, Wendy.
Thanks very much, Anne. You are always providing us with such great insights. We really appreciate you joining our delegation. So without further ado, let's get to the presentation. So the first one, I'd like to introduce Farzian Amindan. He is the Director of Business Development at Verogen Biotech, and I will turn it over to you.
Thank you, Wendy. So let me just share my screen here so we get started. One can see it clearly here. All right, well, thank you, Wendy, and thank you, LSBC, for the opportunity to present. I'm happy to kick off the biotech company presentations by introducing Virogen Biotech. So we are a clinical stage oncolytic virus company focused on novel engineering of the herpes simplex virus to generate strong and durable anti-tumor response against solid tumors. Now, oncolytic viruses have become quite an attractive modality due to its multifunctional ability to directly kill tumors, trigger immune-mediated killing, and also its ability to replicate to infect other tumors. So at Virogen, what we're doing is we're designing better versions of these cancer-killing viruses, and we do so in two ways. The first is how we modify our virus backbones. Now, typically, it's a wild-type virus, they're known to have a strong ability to replicate in tumors, but there's still toxicity concerns. So what folks are doing in the OV space is they're finding ways to attenuate their viruses to address and improve the safety profile. But at the same time, what's happening is they're compromising the replication efficiency by deleting one of the virus genes. So at Virogen, what we're doing is we're designing our viruses to be regulated in a way that only in tumor cells will you have a fully functional virus and with a higher than normal replication and payload expression efficiency. So we call this system the TTDR system. Now, the second way we design more effective viruses is to really arm them with therapeutic transgenes that adds more to what can already be achieved by the virus itself. So when we think about selecting what payloads to incorporate into our virus, we currently focus on three strategies. Now, we know that herpes simplex virus is highly immunogenic. So what we want to do is find ways to really add to that and create a stronger immune storm within the tumor system. So once we actually have this immune infiltration and activation within the tumor sites, there will be some that will be going against the virus. So our second strategy is to find payloads that can help redirect these antiviral immunity into anti-tumor immunity. And lastly, as we all know, the more or less immunosuppressed the environment is, the better. So that's the third strategy of us trying to find ways to reduce the immunosuppressiveness of the tumor microenvironment. Now, we designed our first product to really demonstrate our ability to trigger this immune storm that leads to anti-tumor immunity. Therefore, our first program, VG161, is an attenuated virus, but it's expressing a novel cytokine payload, IL-12 and IL-15, including a PD-L1 blocking peptide. So the intention of this study, of course, for phase 1 is for safety, tolerability, and dosing. But so far, after 11 patients dosed, we don't really see any dose-limiting toxicity, and the drug seems to be safe and well tolerated even at the highest dose. But what we also see are activities to indicate immune cell infiltration and activation. So for example, looking at this one patient here with Merkel cell adenocarcinoma, and that's had already prior treatment of checkpoint inhibitors and chemotherapy, once treated with VG161 at the highest dose, after looking at the biopsy, we do see signs of inflammation signaling over the first couple of days. And you can see here, long-term, it does lead to activation and recruitment of the important immune cell subpopulations that's related to anti-tumor immunity. Now, what's important to know that this is just after a single injection. So what we've seen preclinically and also in other OV companies is that multiple injections per cycle is required to demonstrate this clinical benefit. So our study is still ongoing to determine the right regimen. And we are also having clinical studies to evaluate our viruses in combination with other agents such as checkpoint inhibitors. Now, as I mentioned earlier, on top of selecting the right payloads, a virus with a highly potent backbone also has the potential to eliminate the non-responsive tumor types. So when we incorporate our regulated TTDR system into our virus, preclinically, what we see are three important features. Number one, we do see that compared to the attenuated virus, our TTDR regulated virus still has a comparable safety profile. So what we see is we do still maintain that safety benefit. But at the same time, we also show that our TTDR viruses are actually much stronger compared to the attenuated viruses. And this is seen only after a single dose. You already see observations of durable tumor regression in a really dose-dependent manner, compared to what you see with the attenuated virus. Now, it's not surprising we don't really see any benefit, because we do realize you need multiple injections for an attenuated virus to really see the clinical benefit. However, this is really important to highlight that a single injection is really already highly potent in our TTDR regulated system. Now, what's most exciting for us is that our virus's ability to really kill the distant tumors through immune-mediated killing. So you see here, when we do a study, when we inject the mice in one tumor, there's tumor regression. However, in the non-injected tumor sites, you do also see tumor regression. And also, once we challenge the mice with the tumor, you do see that they maintain the regression of these tumors. So what this data suggests altogether is really our ability to really trigger the systemic effect, which will be of great benefit when we're dealing with metastatic lesions. So we're currently developing several viruses based on our TTDR system engineering and incorporation of different payloads. What's unique about our platform is that we tailor each product for specific populations. So with this tumor agnostic immune precision medicine approach, we're really able to select the patients that will help really guide treatment decision making. So lastly, before we sign off here, we've raised 174 million to date. We do have facilities in Vancouver and Shanghai. In fact, we're really starting a construction of a full-scale GMP manufacturing, which will make us more of a vertically integrated company. We are on track to be a global leader. And when we talk about global leader, we really think about making sure we have a China strategy, since it's an emerging commercial opportunity. So that's why we do have a deep partnership with one of China's largest pharmaceuticals to develop in Greater China. So on top of looking at late-stage development partnerships, what we're looking at is really to find partners to evaluate combination of our oncolytic viruses with other modalities, such as checkpoint inhibitors and cell therapies. And also, we're also looking for partnerships to develop novel oncolytic viruses expressing different types of transgenes and payloads that are synergistic. So if people out there are interested to learn more about our platform and partnering opportunities, happy to chat later on. And also, feel free to connect with me via email. I think my time is up, and thank you everyone for the time.
Thank you very much, and congratulations on your success to date. It's fantastic to see. So I'm now going to introduce our next speaker, Russell McAllister. Russell is the CEO of Vancouver-based Bold Therapeutics. So over to you, Russell.
Thank you, Wendy. So thanks for the opportunity to present. So Bold Therapeutics is a small molecule company in oncology, and we're trying to solve a persistent problem in oncology, in that frequently treatments are ineffective or acquired resistance. And we've discovered an agent that disables a critical resistance pathway cancers, therefore significantly improving outcomes when used in combination with a wide range of other therapies. We're first in class, so we're the first company to pursue this pathway. We're clinical stage. We're in our second clinical study now, a phase 1/2 study here in 6 sites in Canada. We have a very clean safety profile for phase 1, very clean with no hematological or neurological side effects. Driven, you'll see a little bit of the data here in the presentation, but given the slide limitation, only the very tip of the iceberg. We have blockbuster commercial potential. So the data that we've generated thus far suggests that this could and should be used in combination with almost all other oncology agents across a wide range of indications, both solid and liquid tumors. We have robust worldwide intellectual property, including composition of matter issued in the last 2 years. And we have an experienced development team. So BOLD 100, I'm going to talk about the mechanism of action on the next slide, but I first wanted to show you what it does. So we have efficacy, preclinical evidence of that in a wide range of cancers and drug combinations, some highlighted here, and we have new data yet unpublished showing potential in liquid tumor indications such as multiple myeloma as well, which is what was suggested by the literature. So the left panel here is a cisplatin-sensitive gastric cancer model. So this is a model in which cisplatin does well. You can see that in the 8 days of delay in tumor onset growth from treatment with cisplatin. But look what happens when you add BOLD 100 on top of that. It's a massive improvement in the delay in a very challenging model. And the right panel is an ASPC1 gemcitabine resistant pancreatic cancer model, one of the most difficult pancreatic cancer models that exists. You can see a very mild impact from either BOLD 100 by itself or gemcitabine as a monotherapy. Combine them and you get the best results seen yet in this model, 300% extension in survival by combining these two agents. This slide for BOLD 100, it's complex and fascinating. The primary mechanism is inducing cellular stress. We do that 2 ways. We inhibit upregulation of GRP78, which is the master chaperone protein for the unfolded protein response. So effectively in response to cellular stress, cancers across indications upregulate GRP78, improves their protein folding, allows them to survive those conditions, and we selectively stop cancer cells from upregulating GRP78, which drives them toward apoptosis. And in parallel with that, we induce ROS, and that causes DNA damage and cell cycle arrest. So we hit from 2 sides. Again, a very strong safety profile from phase 1. Right now we're combining in our clinical study with FOLFOX, which is one of the nastiest chemotherapy regimens, and our 12 or 13 patients in now with no evidence that BOLD 100 worsens toxicity over FOLFOX alone. And again, we're pursuing a combinational strategy. So we have data showing synergy in combination with a wide range of different classes, a wide range of different solid and liquid tumor indications. We also have orphan drug designations now issued in gastric cancer. Here's a snapshot of what's going on. We signed our first licensing deal with Hana Farm in South Korea last May. Again, we're in a phase 1B study, that adaptive design study that transitions into a phase 2 later this year in just the next couple months. And we're in first and second line gastric cancer and second line pancreatic, colorectal, and bile duct cancers. Data from the phase 1B will be available in the 3rd quarter. Interim data from the phase 2 study should be available at the end of this year, and full data available in mid-2023. We completed an oversubscribed Series A last year, and we're currently raising $30 million in a Series B round. I should also mention we're active in COVID-19. We were one of 4 therapeutics companies selected by the government for therapeutics funding. The same mechanism that we target, GRP78, plays a key role in viral replication. And I'm happy to announce we just put out a press release this morning. We just received another $1 million award for NRCRF for development in that area. We're interested in pursuing a number of additional phase 2 studies in parallel. This is some of our top picks, but we have lots of, and I would add multiple myeloma to that list based on some of the recent data that we've generated. We're also looking for another oncology compound to develop. So if you have something interesting that you think would be a good fit for our team, please reach out to us. And that's it. Thank you very much for the time.
Thank you very much, Russell, and congratulations this morning. That's fantastic news.
Thank you.
So I would now like to introduce Alison Gaw. She's the Executive Director of Business and Corporate Development from CHINOOK Therapeutics. Alison, you're on mute.
Thank you, Wendy. I'm just going to share my screen here really quick.
All right.
Thank you very much. Very happy to be here today to share the story of CHINOOK Therapeutics with this audience, as we're a company founded here in Vancouver in 2019 to focus on developing precision medicine for kidney diseases. Just very quickly, I need to flash the obligatory forward-looking statement slide. So why is CHINOOK focused on precision medicines for kidney diseases, and why do we think now is the time for drug development in kidney disease? Chronic kidney disease is a massive global problem, with almost 10% of the population suffering from some form of kidney disease, which is a huge driver of healthcare spending. And there's been very little progress in kidney therapies, as we still treat kidney disease today as we did almost 20 or 30 years ago. And that's with non-specific medications, such as blood pressure lowering medications or generic steroids. And this is often because kidney disease is grouped into conditions with heterogeneous patient populations with many distinct disease drivers. And this is driven by diagnosed disease driven by symptoms or pathology or histology without a good understanding of the underlying disease mechanisms. But that has slowly been changing these last few years, and CHINOOK is taking advantage of recent developments that help us in our precision medicine approach. With the advent of new tools and technologies like single-cell sequencing, organoid models, and the omics revolution, we're beginning to understand the underlying mechanisms of disease biology and able to deconvolute what is now broadly called chronic kidney disease into the tens, if not hundreds, of specific underlying diseases. Much like the precision medicine revolution that we've seen in oncology, where what was once previously called breast cancer and treated broadly with chemotherapy 25 years ago, is now known as over a dozen different types of breast cancer, each with its own specific type of drug. And lastly, the regulatory environment is beginning to appreciate and recognize important surrogate endpoints like urinary oxalate or proteinuria as the basis for approval. This allows for a clear and manageable development path for a biotech company like CHINOOK, compared to the historically large and lengthy outcomes-based trials that were traditionally required, similar to the cardiovascular field. In the short 2.5 years since founding, we've been busy at CHINOOK building the leading kidney disease company, with a full pipeline led by our 2 clinical programs, atrasentan and BION-1301. Atrasentan is a highly potent and selective endothelin A inhibitor. We licensed this program from AbbVie, who had previously studied it in over 5,000 CKD patients. However, early on, the research team at CHINOOK discovered that endothelin A specifically and the ET1 system is very important in IgA nephropathy and other proteinuria glomerular diseases, and drives progression in these diseases through a number of unique mechanisms. Based on these findings, we've initiated the phase 3 ALIGN trial in IgA nephropathy and phase 2 AFFINITY trial in other glomerular diseases. We expect data from these trials starting in 2022. Last fall, we completed a merger with Aduro Biotech in Berkeley, California, which not only brought with it our NASDAQ listing and significant financing, but also the program BION-1301, an anti-APRIL monoclonal antibody. We're very excited about this program as it has the potential to be disease-modifying for patients with IgA nephropathy. APRIL is a TNF family cytokine that is involved in B-cell signaling and IgA class switching. It drives the survival of IgA-secreting plasma cells and the production of IgA, including the galactose-deficient IgA form that is the pathogenic driver of IgA nephropathy. Last year, we presented PK and PD data from the phase 1 trial of BION 1301 in healthy volunteers. And just yesterday, we presented the initial patient data at the ERA EDTA conference. The data demonstrates durable reductions in free APRIL, IgA, and Gd-IgA1 levels in both healthy volunteers and patients with IgA nephropathy. We will continue to present additional data from patients later this year. For our preclinical pipeline, we're also very proud of our first in-house developed program at CHINOOK, CHEK336. This is an oral small molecule that inhibits LDHA, a key enzyme in oxalate production. More than 80% of kidney stones are due to excess oxalate, which can lead to kidney damage and failure. We're developing CHEK336 for primary hyperoxaluria, a group of ultra-rare genetic diseases, which, if left untreated, can result in kidney failure for patients as soon as their early 20s. With its oral small molecule form, we believe that CHEK336 also has the potential to treat all disorders of excess oxalate. Underpinning all of our efforts in kidney diseases is our precision medicine approach. We're focusing on rare and severe chronic kidney diseases where we can use the greater understanding of disease biology and mechanisms to design novel and targeted and differentiated drugs to study in well-defined patient populations and conduct efficient clinical trials with biomarker or surrogate endpoints. We have collaborations with leading academics like Ben Humphreys at the Washington University of St. Louis, one of the leading pioneers of single-cell sequencing. And through a partnership with Evotec, we have access to the Nurture Kidney Biobank of over 5,000 patients where comprehensive pan-omics analyses are underway. To summarize in graphical form again, the pipeline for CHINOOK, we have our clinical programs with the ongoing phase 3 aligned trial in IgA nephropathy and phase 2 affinity basket trial in other glomerular diseases for atrasentan, and an ongoing phase 1/2 trial for BION 1301 in IgA nephropathy, followed by our preclinical program CHEK336 and a host of other research discovery programs. CHINOOK has global commercial rights to all of our pipeline programs, and in the future, we may be seeking commercial partners for our programs in select geographical regions when the time is right. Additionally, we continue to evaluate opportunities to add kidney disease programs to our pipeline. CHINOOK is in a strong financial position, and we have guided a cash runway through the first half of 2023 based on current business plans. It is an exciting time for the company, and we look forward to progressing our pipeline towards commercialization to make a difference in the lives of kidney disease patients. Thank you.
Hi, maybe Wendy is having a difficulty getting back on today. I can kind of move to introduce our next speaker, which is Mohammed Qadir, who is the President and Chief Scientific Officer of Fusion Genomics. Mohammed, if you're here, if you want to pop on.
Hello, everybody. So thank you very much to the organizers for allowing us this opportunity. And we are Fusion Genomics. Without any further ado, this is the disclaimer. So we are in a pandemic. Imagine that we could have identified this pandemic before it could have started. Imagine that we wouldn't have to chase all the variants after the fact that they had emerged. Imagine we could have stopped this pandemic, or we can stop any other pandemic. This is not the future, this is the present, and this is one of the reasons that Fusion is here. How is that possible? That is possible because what Fusion has is a targeted metagenomic assay which can work on any biological sample. It's a one test which allows you to test for any pathogen that you're interested in. either one or hundreds of pathogens at the same time. The other thing is that the assay, or our technology to capture the genomes of viruses, bacteria, parasites, and fungi, is based on evolutionary information, which means that it doesn't matter how much the virus has mutated, or if it's a novel virus, like in the case of Corona 2, where it has jumped from a zoonotic species into humans, we can still detect it. So what happens when you have a platform technology like that? You're not chasing things. Everything happens in real time. From the time that you identify that first patient, that patient has something novel, you can share that information with all the stakeholders around the spectrum, which is your public health agencies, researchers, policymakers, and vaccine and therapy developers, because at the end of the day, this is a genomic test. So these are bold claims, and Fusion has been validating these bold claims with all these partners that you see on the screen. The main aim of Fusion is that currently we are still pre-commercial, but the main aim of Fusion is that when we enter into the market with a new diagnostic assay, we just don't want to be better than the predicate. We want to be the gold standard. And that's what Fusion is all about. Now, I'll share very little amount of data from one of these partners and to explain you the claim. So this is a patient from a very early days of the corona or the SARS-CoV pandemic in Canada. And we tested this patient with a version of our assay, which was designed in 2018. And you know that in 2018, SARS-CoV-2 did not exist as a pathogen in humans. It has not yet jumped from the zoonotic species. Yet, using the assay, we were able to capture the genome of it, and we could instantly identify that this virus, at that time, they had already published the genome of the coronavirus from Wuhan. So it instantly aligned the genome from this patient and saying, this is most closely related to this genome. But if we didn't have that information, if you see on the screen, the number one is the Wuhan, but underneath it, it is matching with a bad coronavirus. So it would have told the clinicians, the researchers, policymakers instantly that you have a novel zoonotic jump into humans. This platform and this kind of technology is what also led to the early use or the trial use of the platform with various government agencies around the world. And in Canada, it was announced last year by Minister Baines. But Corona came in, but we existed before Corona. So what was really the problem that Fusion was addressing? The problem that Fusion addresses are the undiagnosed infections in a hospitalized patient. As you can see on your screen, all major categories of infections have undiagnosed patients. From a billing standpoint, just keep into mind that the initial market is for the 2 midnight rule and DRG weight billing when it comes to US. And as you can see, all of this are on every category is that undiagnosed patient. To think of it, think like this in simple terms. That very first patient in Wuhan, in that hospital, that was an undiagnosed respiratory infection patient. So in that context, the clinicians instantly recognized this is an upper respiratory infection, but all their tests were failing. So that becomes an undetected or undiagnosed infection. This is a major problem. Just in 8 years between 1998 and 2006, this problem cost about $27 billion a year, every year in the US alone. And that number is growing. The other ironical part is that this cost comes from only or less than 15% of the patients. And this is the Fusion's target market. This is a busy slide. What is Fusion's real value proposition? As opposed to conventional microbiology and then the use of whole metagenome sequencing for companies like carriers, ID by DNA and everything, the earliest they can give this assay is about as about at 50 hours. And they can only provide it as a service because of the complexity. What Fusion has is a lab on a chip which can go straight into the hospital, give you your percentage of diagnosed at 24 hours. Just to give you an idea of the market, for every 1,000 beds, there are 500 of these high-risk patients. By that magnitude, there are about 0.4 million cases. So just the initial market is about 200 million. Now, if you move this assay as a frontline assay, then it expands into a multi-billion-dollar market. And that's where Fusion is headed. Thank you, everybody. I think the time's up.
Thank you very much, Kedar. Fantastic developments in fusion genomics. We're excited to be able to provide you with the opportunity to present here, and hope you make lots of connections through the week. Our next speaker is Paul Brennan, and Paul is the president and CEO of NerveGen.
Hey, Paul, if you need me to share, I'm happy to do so, and you can cue me. Let me know. And you're just on mute, by the way.
You're on mute again, though.
But your presentation looks good.
Okay, let's try this. How's that? Is that working?
That's great.
Awesome. Okay, so NerveGen is a neurosciences company, clinical stage, and we're focusing on developing products for indications where there's nerve damage. So our technology is a nerve repair technology. Our lead indications are spinal cord injury, multiple sclerosis, and Alzheimer's disease. So our presentation has forward-looking statements. If you need to look at the risk factors, please refer to our CR filings. We are a public company. So our technology comes out of the labs of Dr. Jerry Silver at Case Western Reserve University. And Dr. Silver was interested in spinal cord injury and understanding why, when you have a spinal cord injury, the nervous system doesn't repair itself. And he came upon the discovery of a class of molecules called chondroitin sulfate proteoglycans, or CSPGs. These are macromolecules with a core protein and glycoaminoglycan side sugars. They're present in healthy tissues as a structural component and also in early development and neural guidance. But they also play a critical role when there's neuronal damage. The CSPGs are upregulated significantly. And they do that to protect the site of damage to contain the damage from further damage, to basically restrict the damage. However, unfortunately, they also have a long-term side effect. In the long term, the CSPGs inhibit axonal regeneration, they inhibit plasticity, remyelination, autophagy, and they also inhibit the microglial cells, or they promote an inflammatory phenotype in microglial cells, which inhibits repair. Evidence of CSPG upregulation has been seen in multiple diseases, not just spinal cord injury, where Dr. Silver was interested. We see them in humans in multiple sclerosis, Alzheimer's disease, stroke, Parkinson's, peripheral nerve injury, and also seen in animal studies in ALS and traumatic brain injury. basically any time that there's damage, whether it's acute damage or chronic damage, you see CSPGs upregulated. Dr. Silva's other important discovery was the identification of a receptor called protein tyrosine phosphatase sigma, or PTP sigma. PTP sigma is the predominant receptor by which CSPGs are having their inhibitory effect. Basically, in the presence of CSPGs, PTP sigma has a number of downstream signaling pathways that will inhibit axon growth, inhibit plasticity, inhibit repair, inhibit autophagy. So Dr. Silva's next great discovery was to identify a number of inhibitors that would inhibit the activity of PTP sigma. So by inhibiting the inhibition of repair, Dr. Silva's peptides were allowing or promoting repair. It's these peptides that are the basis of our technology and our lead compound, NVG-291, which is now in phase 1 clinical trials. So the results that we're seeing with NVG-291 are really quite unique. Unique in terms of the pattern that we're seeing and the magnitude of effect and the breadth of effect. So typically, when you're looking at one receptor, you see one receptor, 1 or 2 pharmacological responses, and maybe 3 or 4 different diseases to which it's applied. PTP inhibition has minimally 5 different pharmacodynamic effects that we've seen in preclinical studies. We've seen nerve regeneration, remyelination, enhancement of plasticity. We've seen them change the phenotype of the immune cells and enhancement of autophagy. What this means, of course, is, or what's important is that you have a functional response. And this is where we see the breadth of response that we have. We're seeing improvements in motor function, both fine and gross motor function, sensory function, bladder control, heat detection, autonomic function, sensory function, heat detection, and vision, autonomic function, bladder control, pacemaking, and cognitive function. So just to give you an example of the magnitude of effect we're seeing, I'm going to show in the next slide an example of a spinal cord injury that's been given to a rat. And you can see the hind legs of the animal here. The animal does not have any control. This is a control, no control of the hind legs, not able to bear weight. Now, if you look at the treated animals, you'll see on the right, look at the hind legs, the coordination between the legs, the ability to bear weight. This level of improvement is unprecedented in pharmacological therapies of spinal cord injury. When you look at the scale of improvement, the magnitude of what we're seeing is quite unique. This is not an indication where you put lots of molecules in and you see lots of positive results that don't translate to humans. This type of response is really quite unique. In terms of NervGen's path forwards, as I mentioned at the very beginning, we are in phase 1 studies in healthy volunteers. Those phase 1 studies are in the single ascending dose cohort. We plan to initiate our multiple ascending dose cohort in the second half of this year. Those will read out early 2022. And in mid 2022, we intend to initiate our phase 2 studies in spinal cord injury, multiple sclerosis, and phase 1b in Alzheimer's. And so with that, I'd like to thank you for listening to the presentation, and hope everybody has a great bio or virtual bio. Thank you very much.
Thank you very much, Paul. I'm now going to introduce our next speaker, Kareem Lalji. Kareem is Microbiome CEO, and it's great to have you with us.
Thank you. It's a pleasure for me to be here. And I know Tim is actually going to be projecting my slides for me. We had a few technical issues, and it's in front of me right now. So it's a pleasure to be here today. As I said, I'd like to thank the Life Sciences BC team for inviting Microbion. To start with, I just want to recognize my team, which is on the next slide. We are a pretty small team, but we do a lot of great work. And half of the team is here in Vancouver, BC, and the other half of the team is sitting in the US. So we are one of these companies that actually works on both sides of the border. And our team has a lot of experience from many companies in BC, as well as companies in the United States. As I move to the next slide, I'd like to frame what we're doing at Microbion. we are developing a new class of drugs to treat basically multi-drug resistant infections. And the key differentiator that we have is we are able to eradicate biofilms. And there's been a lot of work done with biofilms, and what basically has been shown is that the most multi-drug resistant pathogens are also the most prolific biofilm formers. So there's a real interplay between biofilms and resistance. And we are able to effectively eradicate the biofilms and the bacteria at equal potent doses. And most antibiotics that are available today are completely ineffective at treating biofilms. And even if they are, they're at doses at such a high level greater that are needed to kill the biofilms that they move into the unsafe territory. So yeah, as we move to the next slide, I'd like to speak a little bit about pravibismane. So pravibismane is what we believe a disruptive therapy, and it's a first-in-class therapy. We have been granted the bismane stem by the WHO, and this is the first drug in this class. And what's really interesting about pravibismane and this class is as I mentioned, the ability to eradicate biofilms. We already have dosed over 300 patients with this product. And ultimately, the difference that we have is the mechanism. We effectively shut down the energy production of the cell. As we penetrate the biofilm, we get into the bacteria, we shut down the energy production. The bacteria actually keeps the biofilm alive, and therefore the biofilm collapses because there's no energy to keep it going, and then the cell collapses. So it's a very targeted way of eradicating both the bacteria and the biofilm, and it's a new mechanism, and the WHO has recognized us as a new class, which I think is very important. As we move to the next slide, and I won't spend much time on this, but we are a very broad spectrum agent with very good potency. We eradicate and are able to address all of the priority pathogens identified by the US CDC. As I move to the next slide, I would like to just share with you our strategy. Being in infectious diseases, being in the anti-infective space has not necessarily been that rewarding historically for investors. We recognize that it's been a problematic space, but we are doing something different in the way we are developing our product. While we see a lot of benefit from a systemic product, and we will do that at some point, we are initially focusing on targeted local administration. So I'll get into the specifics of how we're doing that in a minute. But going to a targeted local administration allows us to get to greater efficacy locally, greater safety because we're not actually going to, through the systemic circulation. A topical approach, for example, to treat a diabetic foot ulcer infection is very different than a systemic. We also have a reduced risk of resistance because that is one of the concerns that clinicians have. If you're not having systemic administration, if you're going local, there's less risk of resistance. And we also have favorable reimbursement by going targeted. By not being a hospital-based product, you don't have the hospital controls. And that is really also an important part of the approach commercially that we're taking in development of our product. As we move to the next slide, it just highlights the three areas that we are focusing on. We have already run clinical trials in diabetic foot ulcer infections, a very large market. The US sales opportunity is considered to be about $1 billion. We have an orthopedic program that's also in the clinic, also a large market. And we have an inhale program that will be entering the clinic in the next 6 months. And that is an orphan targeted use. So basically we have orphan designation already for cystic fibrosis related infections, and we're also looking at NTM infections. As we look at this next slide, this is simply results of our study in diabetic foot ulcer infections. This was only 3 weeks of treatment, and we saw dramatic reductions in wound size. The FDA has indicated to us that we can go into a pivotal study next. We have decided to do one small proof of concept because we saw these results with only 3 weeks of dosing. We will be dosing for 12 weeks, which is standard, and we are looking for reductions in amputations as well as closure of wounds as a signal that we will then move into our pivotal studies with. So we're very close to actually moving into our pivotal study, And because we have something called QIDP designation, there is also an opportunity to be approved by the US FDA with one single pivotal study, which we think is a really efficient way to getting to market as well. As we move to the next slide, this is basically a summary of the work we're doing in the inhaled space. And there's a key competitor out there that's developing a drug for NTM. We have demonstrated superiority in vitro to that compound, and we also have other elements of key differentiation. And the reason I raise this is that you have a weak profile agent in an agent called Arikayce, and that has driven a $3 billion market cap for this company. And we believe we have a better agent, a superior agent, basically a better mousetrap, and we believe we should do better. But the point I'm making is that an anti-infective local administration has generated a very nice, a market capitalization and is a good return for investors. As we go to the next slide, I just want to comment that we've raised a lot of money at this company, over $50 million, and just in the last 18 months, we've brought in $20 million of non-dilutive capital to support primarily our inhaled program, but also our DFI program. And we've also brought in dilutive capital, and I'd like to recognize a BC group, Quark, for being part of our investor base. And as I go to my last slide, I just want to lay out that we are raising a fair amount of money, and this is our clinical program that we're targeting. I won't get into the details. I look forward to speaking to investors on this afterwards. And just want to thank everyone for your time here today. But again, a new class of drugs, very unique, providing very important solutions and we have comparables that demonstrate success in these areas, can generate very good market capitalizations for the company. And we are basically in the clinic for all three indications as of 6 months from now. Thank you all for your time. Sure.
Thank you very much, Kareem, and that was a fantastic presentation. I'd now like to welcome Scott McMillan, co-founder and CEO of Exco-Tech, to present. Over to you, Scott.
Thank you, Ryan. Here, let me set up. Thank you for the opportunity to present today. Exco is a health and performance company that has developed cutting-edge motion, cognition, and biometric health technologies. I am here today to present Recon Health, our new joint venture with Atlazo, a leading semiconductor and AI company out of San Diego. Together, we have developed an AI supercharged device with built-in insights for chronic disease patients to best inform their clinician virtually and to better manage their own health with medical-grade data. Recon is led by Jim Miller, Kareem Arabi, and myself. I have been developing and marketing health and performance products throughout my career, including many years with the Adidas Innovation Team. Jim is a serial healthcare entrepreneur, having been a founder of QLT, NX Techmera, Alon, and NDI Capital. And Kareem was VP at Qualcomm, responsible for R&D. We all share the belief that in healthcare today, there is a unique opportunity to advance that diagnosis and treatment of chronic diseases by using data to dramatically improve patient outcomes. As you probably are aware, virtual care, in particular telemedicine, blossomed during COVID-19, growing 10x. What is interesting is there is still 10 times the amount of healthcare spend that can be virtualized, if it can be better enabled. One of the most widely recognized limitations in virtual care today is that critical vital sign data isn't available to doctors, since patients generally aren't well equipped with medical devices at home. Yet key vital signs, along with other measures, are critically important to diagnose and treat acute and chronic diseases, including cardiovascular, respiratory, and neurological conditions. Interestingly, 50 to 80% of primary care could actually be done given the right tools were ready available at home. Our solution is to provide Recon Health's membership that comes with a virtual care patch and access to multiple continuous health monitoring insights based on SpO2, heart rate and heart rate variability, respiration rate, core body temperature, and activity readings. We also are adding ECG, speech, and blood pressure. And all of this can be shared with doctors, which I'll talk about in a minute. Let's first break down the key features of the patch. The virtual care patch comes with exclusive health system on a chip technology, including embedded AI. We are leading the way in implementing AI on a medical device that fuses multiple measurements at the source, curates the data, and provides analytics with high accuracy, all of which are enabled by the chip's high energy efficiency. We are providing a patient-first experience so that they have access and control of their data and can use the insights to better manage their health. And that includes the option to share the data seamlessly with multiple virtual care providers. Now, today, data is king in a lot of industries. Healthcare is no different. But really, it is the insights that are king. Since patients and doctors don't want mountains of data to sift through, and even if they did, they don't have time. Recon's patch, with its edge computing embedded AI, enables actionable insights to be driven from high volumes of data that was previously unattainable with devices on the market today. But what does that mean to the patient and clinicians? What it means is, it means automated logging of multiple vital signs. It means automated analysis to save patients and doctors time. It means high resolution data and medical grade accuracy, and data available in real time, along with edge computing privacy advantages, and extended battery life to avoid constant recharging. This is why Recon's patch is not just another wearable. With research and commercial partners, we are working on continuous respiratory analysis, detection of cardiovascular events, fever response, sepsis, fall prediction and detection, tremor tracking and PD, and speech analysis for insights into cognitive decline. Those solutions and Recon's core offering are filling an important gap in the market. On one hand, you have lifestyle wearables, which fit loosely on your wrist and simply aren't trusted by doctors. While on the other hand, you have medical devices that are designed for clinicians and are convenient, easy to use for patients, and don't readily provide them insights and information. Recon provides a patient-first experience with outcome measures for their particular condition with that medical-grade accuracy. To highlight these differences, I'll compare now Recon's patch to one of the leading devices in the market today. The Zio patch by iRhythm is used to detect arrhythmias in patients. It's quite expensive, non-reusable, and provides a 4+ week turnaround before the patient sees any data. Whereas the virtual care patch will provide ECG analysis along with multiple vital signs and provide these results and insights immediately to the patient and doctor. And the distributed computing approach is very cost-effective, resulting in consumer-friendly pricing and quick market penetration. Our go-to-market plan is underway, including validation of the patch with leading healthcare organizations across multiple chronic diseases. We will drive awareness through direct marketing and providing educational marketing content to clinicians, including how they bill for services using existing reimbursement codes. Lastly, we simplify access to the product through Recon's e-commerce site. Patients will be driven to the site by their doctor, insurance provider, employer, or from Recon's digital marketing. In terms of revenue and market size, we are displacing $5 to $20 billion slower moving companies such as iRhythm, Biotelemetry, Livongo, and more. And we expect a modest 2% penetration into the chronic disease market within 5 years. To date, we've established over 9 patents, fully implemented the novel chip technology, and validated the vital sign measurements, and are now raising capital. To deliver a first-class experience to patients and their caregivers with technology that can truly help them improve their health, we are raising $5 to $10 million USD to finalize the product, do regulatory filings, and execute on marketing and sales. This will be milestone-based financing using our experienced teams that we already have in place. For more information, please feel free to reach out. Thank you.
Great, Scott. Thank you very much for your very insightful presentation, and thank you again to everyone for their great presentations. Apologies that Wendy is just having some internet problems, so I just wanted to give some closing remarks on behalf of herself, myself, and all of our team here at Life Sciences BC, including Tim Hennessy, who's our Manager of Events. Would like to thank the Honorable Ravi Kalin for his opening remarks, and to our speakers, Farzian Amadoun, Paul Brennan, James Hatton, Alison Gaw, Russell McAllister, Scott McMillan, Mohammed Kadir, and Ann Woods. Also, a special thank you to our sponsors, to our event sponsors, Ferris and Silicon Valley Bank, our event supporter, the Government of British Columbia. And this event would not be possible without the generous support of all of our sponsors. So I'd like to thank everybody for attending today, and we are looking forward to seeing everybody again in BioConference 2022 in sunny San Diego. And the last note, just wanted to remind everyone that we have our BC booth at our Canadian Pavilion, and you'll see all of the companies listed here. And also on the other slide there is all of our annual sponsors. And so if you're interested in becoming an annual sponsor or becoming a member, please reach out to us, as we would love to have all of your support. And now we will move on to networking. So, and also to encourage you to reach out to all of the companies that presented today, as well as this full list here, and set up one-on-one meetings for the very exciting and busy week of BioDigital. So thank you again, and look forward to, we're now going to do networking for about the next half hour. So I will look forward to seeing you on the floor, and all of our speakers have individual tables there. So thanks again, and thanks for joining us today.